our next speaker is Olivier Parent. Okay, stage is yours.
Hi everyone, thank you for the introduction. So yeah, my name is Olivier Parent, and I'll present to you my new project on Voxel-wise normative models for microstructure. Okay, I guess this doesn't work. I think the change slide doesn't work. Oh, okay, now it works. So to estimate the brain tissue microstructure, most ground truth methods are only available ex vivo, so things like histology and two-photon. But with microstructural MRI, we can derive in vivo proxies, and so we can now detect brain pathology in vivo, which has the potential to derive biomarkers which are sensitive to diagnosis and prognosis for certain diseases. So here I'll present three microstructural biomarkers across disorders. So first, for multiple sclerosis, this study by Lopez Soli et al. Reported that fractional anisotropy in certain right matter regions were predictive for future multiple sclerosis disability. And so they conclude that these results provide relevant information for developing more accurate predictive models that could help therapeutic strategies. Stop working again. Okay, so for Alzheimer's disease, this study by Spotorno et al. Reported that mean diffusivity in certain cortical gray matter regions, which are early amyloid regions, were predictive of future Alzheimer's disease progression. And so again, they conclude that these results support the possible role of metrics derived from diffusion MRI in tracking early changes in the AD continuum. For Parkinson's disease now, the study by Sun et al. Reported that quantitative susceptibility mapping values in certain subcortical gray matter regions had a good diagnostic accuracy to diagnose Parkinson's disease. So again, they conclude that this technique has the potential to be an objective neuroimaging biomarker for the detection of the prodromal stage of alpha-synucleinopathies. So you might see where I'm going with this, but despite showing a lot of potential for diagnosis and prognosis, none of these biomarkers have actually been translated into clinical settings. And so this led me to ask myself the question, why is clinical translation so limited for these microstructural biomarkers? And I think, in my opinion, one of the main roadblocks is about how we visually present these raw microstructural maps. So for example, here you see the FA map for a 39-year-old female who has multiple sclerosis. And so I don't know about you, but I don't see anything that's striking about the FA map, no pathology that is easily detectable. If we compare side to side with the FA map from a healthy 49-year-old female, we can see maybe perhaps a lower FA values in the person with multiple sclerosis, but obviously this is not very optimal to detect pathology, and it's not something that would happen in a clinical setting. So I think there's a lot of problems with how these maps are presented. So first, the contrast is mostly driven by the anatomy, but not the pathology. Second, it's hard to disentangle normal age and sex-related processes from pathological processes. And third, I think the interpretation of raw microstructural values is very unintuitive. So for example, is an FA value of 0.35 good or bad? The answer is that it depends on the region, it depends on the age, sex of the participants, so it's very unintuitive. And so the solution I'll be presenting today is instead to derive what I call abnormality maps, where each voxel is expressed at the number of standard deviation away from a healthy microstructural norm. And so for this example here, we can now clearly see that the subject with MS does have a very low FA in a lot of white matter regions, which is now very easily identifiable. So first, to get these maps, I needed to establish healthy microstructural norms. So I did that using the data from 17,500 healthy participants from the UK Biobank. So these participants didn't have any diagnosis on endocrine, metabolic, behavioral, nervous, and circulatory system across their lifespan, but we did make a few exceptions. We had access to these 7 microstructural markers, which I broadly categorize as fluid sensitive, fiber sensitive, and myelin and iron sensitive. We also did a lot of a few steps of image processing in the UK Biobank. So we did tissue segmentation using the Bison tool developed by my co-supervisor, Masadadar. And we also did multispectral registration using both T1 weighted and FA maps to a customized UK Biobank template in order to really accurately get voxel by voxel correspondence across subject, which was really critical for our application. Then we derived Voxel-wise tissue-specific normative models. And what I mean by tissue-specific is that if we take this voxel, for example, sometimes it will be in the white matter and sometimes in the gray matter for different subjects because of registration errors or simply brain variability. And so these will have drastically different microstructural values. And so we fit what we call tissue-specific normative models. So for example, here, one model for gray matter and one model for white matter. And so we do that at every voxel in the brain using Bayesian linear regression with third-order B-splines for age and sex as covariates. And this was all done with the PCN toolkit from André Marquand's group. So, and I really want to thank them because their documentation is very thorough and it made it really easy for me to implement this toolbox. So thank you if you are here. Yeah, so this whole process resulted in averages in age, sex, and tissue-specific averages of healthy tissue, which can now use to z-score individual microstructural maps. So in my results, I'll first present case studies visually comparing raw and z-score maps in common space. And then I did a group analysis looking at the proportion of focal abnormalities defined as absolute z-score of more than 2 and comparing people with a certain diagnosis with healthy controls. So for the first case studies, here we have 2 subjects with multiple sclerosis with, as you can see on the flare, somewhat similar amounts of white matter lesions, maybe a little bit more for the subject on the top. If we look at the raw microstructural maps, you know, I think it's really hard to, at least first glance, detect any differences between the 2 subjects. But if we look at the z-score maps, it now becomes very clear that, in fact, these 2 subjects have very different abnormality patterns. So if we focus here on the MD and ISOVF, we see that for the subject on the top, the abnormality is really contained within the lesions, while for the subject on the bottom, the abnormality is really widespread across most of the white matter. And so this could indicate early inflammatory processes that could lead to the formation of new lesions, which is now very easily identifiable with the z-score maps. So at the group level, these graphs I'll be showing kind of represent the disease signature of focal tissue abnormalities across markers and brain regions. And so as we would expect for multiple sclerosis, we see a lot more abnormalities, focal abnormalities in the white matter, which is picked up by MD, ICVF, and FA. But we also see more focal abnormalities in the gray matter, in subcortical and cortical gray matter, mostly picked up by MD and ISOVF. So now this 58-year-old female was diagnosed with Alzheimer's disease 3 years after the MRI visit. And so I kind of challenge you to maybe try to detect the pathology or any signs of Alzheimer's disease. Maybe I'll give you a few seconds. There's a lot of information there, but time is flying. So I'll give you the answer. So what I find particularly interesting here is that the MD and SOVF in the medial temporal lobe regions is particularly elevated in this subject. And so as we saw in the biomarker study I presented in the introduction, this could really represent early Alzheimer's disease-related pathology. At the group level, we really support these findings. So we see a lot more focal abnormalities in the cortical gray matter for MD and ISOVF, but we also see more abnormalities in the white matter, subcortical gray matter, brainstem, interestingly picked up by QSM, and also cerebellum gray matter. Next, the 75-year-old male was diagnosed with Parkinson's disease 2 years after the MRI visit. So again, maybe I'll leave you a few seconds to try and see for yourself if you find anything interesting. But what I find interesting here is that the T2 star is really low and QSM is very high in these subcortical gray matter regions, which could clearly mean accumulation of iron, which, as we saw, is really a hallmark of Parkinson's disease. At the group level, we somewhat confirm these findings with a little bit, few differences. So we do see more focal abnormalities in the subcortical gray matter, but we see that these are mostly significant for MD and ISOVF instead of T2 star and QSM. So these 2 markers could be even more sensitive to PD pathology than we previously thought. So now for my conclusion, so I think these z-score maps have a lot of advantages versus the raw microstructural maps. So they provide convincing visual evidence that is really easily interpretable by humans and clinicians potentially. They remove the spatial contrast driven by the anatomy. So what we're left with is the contrast driven by the abnormality, which is what we're really interested in to detect pathology. They also help disentangle pathological and normal aging processes. And also importantly, all images are in the same scale, so you can now compare across images very, very easily. Few limitations, so I think obviously the z-score maps are only as good and as biologically interpretable as the raw maps. So if you have artifacts in the raw maps, you'll also get them in the z-score maps. There is a few sources of added noise with the normative modeling, registration, and segmentation, but I think these are pretty well mitigated. And obviously, to really apply this technique in clinical settings, we'll need to handle side effects, but I think this is doable. There's a lot of methods that have been presented in this conference, so I think this can be done. Yeah, I'd like to thank all my lab and my collaborators for their help. If you like the brain images I presented here, they've all been generated with my colleague Johan Yi ggplot add-on, so you can follow the QR code link here if you're interested. And I'll be on my poster this afternoon if you want to come chat, then I'll be very happy to see you. Thank you.
Thank you. We have one question from the app. Can you repeat the name of the toolkit? It's on the screen.
The name of what?
The toolkit.
Toolkit, it's PCN toolkit. Yes.
Any questions from the floor?
Can I ask a question? So what do you think is the interpretation between the different sensitivities of the different parameters to different conditions? Like sometimes it's the MD and the ISOVFs, and sometimes it's the SWI.
Yes, so I think MD and ISOVF, as we saw, are very sensitive to a lot of different pathologies, and so I think this is interesting. So I think it can pick up inflammation, as we saw in the literature. There's been evidence for that, and also edema. So I think these are two processes that are very, very clearly there in a lot of pathologies. And I think T2 star and QSM, it also matters in the quality of the data. And I think in UK Biobank, the diffusion MRI is a lot higher quality than susceptibility MRI. So I think maybe with the added noise there, you could maybe less easily detect pathology with the SWI.
Okay, thank you.
Thank you.
We got another question. Still have time. We got another question from the audience. Would it introduce new challenges for the clinic translations, as neuroradiologists are not used to such images?
Yeah, I think certainly, like to properly interpret them, you'll need to know what MD, ISOVF, all these acronyms mean. So there's definitely going to be a training there potentially, but I think it's first like we need to show that they provide enough information that could maybe motivate them to learn these new techniques. So
maybe I could add on another comment. To be able to use this in the clinical setting, how do you think about the batch effect?
Batch effects, yeah, it's going to be very crucial. I think that's the main problem that we're going to be facing forward to implement these in clinics. But I think there's been work shown by Andrew Marcan's group that you can use, for example, transfer learning to apply these normative models to new data sets, and you need maybe 20, 30 controls to, and they've shown that that's enough to do transfer learning. So I think that's doable.
Thank you.
Thank you.